Hello and very good morning to everyone. Welcome back once again to the fifth day of 1 week International Faculty Development Program on Herbal Medicine, Research and Global Health, a Therapeutic Perspective, organized between 9 to 14 Feb by Adulteration Prasarak Mandal, dedicated to religious participants. The purpose of education is to replace empty mind with this beautiful quote. Let's begin our new day of this International Faculty Development Program. I hope you are enjoying our different knowledge boosting sessions and getting benefits from them since 4 days and now ready to attend the session for today. Dear participant, in the present era, universal trend has been shifted from synthetic to herbal medicine, that is return to nature. As per World Health Organization, there are three kinds of herbal medicine: raw plant material, processed plant material, and medicinal herbal products. Herbal medicine are complex chemical mixture obtained from plant which is widely used in healthcare in both developed and developing countries. It is no wonder that the world's one-fifth of population is using traditional medicines for the treatment of various ailments. However, one of the major drawbacks in the acceptance of herbal medicine is the lack of standard quality profiles. Due to the complex nature of inherent variability of chemical constituents of the plant-based drug, it is difficult to establish quality control parameters. Ladies and gentlemen, the analysis and quality control of herbal medicines are moving a step ahead towards an integrative and comprehensive direction in order to tackle the complex nature of herbal medicine. High performance thin layer chromatography, that is HPTLC, is one of the sophisticated instrumental techniques for qualitative and quantitative analysis of the herbs and herbal drugs. So, for to know more aspect regarding use of HPTLC in identification and purity checking of herbal drugs, we have a very eminent personality in our expert panel today. Our resource person for today's session is Dr. Uday Arvind Devkate sir, Associate Professor, Government College of Pharmacy, Aurangabad, who will orient us about role of HPTLC in identification and purity checking of herbal drugs. I would like to give a heartiest welcome to Dr. Uday Devkate sir. Welcome sir, and thank you very much. You have accepted our invitation
Thank
and
you.
spared your precious time
Thank you.
with us. So before we move towards the session, I would like to introduce
Thank you, ma'am.
Dr. Uday Devkate sir in brief. Sir has completed their diploma in pharmacy from Kamla Nehru College of Pharmacy, Aurangabad in 1987, B.Pharm from Pravara Rural College of Pharmacy, Pravaranagar in 1991, and M.Pharm in Medicinal Natural Products from NDMVP's College of Pharmacy in 1993, Nashik. Sir has completed PhD in Pharmaceutical Sciences from Civil Private Bhavati University in 2012. Sir has completed Industry Program in Pharma Quality Assurance and Quality Control Bioinformatics Institute of India, Noida, UP in 2013. He has appeared for postgraduate graduate diploma in educational management and administration. Sir is currently working as an associate professor in Government College of Pharmacy, Aurangabad. Sir has done research work on isolation and identification of Malkangani oil in M-Farm and isolation, chemical characterization and screening of some Indian medicinal plants for hypoglycemic activity in PhD. Sir has 29 years of huge experience in teaching. He has guided two students for PhD work. Sir has published two patents on his credit. He is an approved PG, UG and PhD teacher of Dr. Babasaheb Ambedkar Marathwada University, Aurangabad and PhD guide of Jawaharlal Nehru Technological University, Kakinada, Andhra Pradesh. Sir is a life member of Association of Pharmaceutical Teachers of India and is Indian Institute of Technical Education as well as it is a registered pharmacist. Sir is a member of examination committee and panel of examiners of different universities, University of Pune, Dr. Babasaheb Ambedkar Marathwada University, Aurangabad, Swami Ramanand Tirth Marathwada University, Nanded, Shivaji University, Kolhapur, Sant Gadgibaba Amravati University, Amravati, Rashtrasant Tukaroji Maharaj, Nagpur University, Nagpur. Sir is worked as a paper setter for multiple choice question paper setting for state level entrance examination conducted by Government of Maharashtra for admission to M.Pharm post from year 2006 to 2015. Sir has handled and worked on instruments like UV visible spectrophotometer, IR spectrophotometer, HPTLC, HPLC, auto titrator, spectrofluorometer, GCMS, etc. Sir has participated and worked as a resource person in many seminar, conferences, and staff development training programs. Sir has given his contribution as subject chairman for syllabus framing committee constituted by Dr. Babasaheb Ambedkar Marathwada University, Aurangabad, and worked as a resource person in the refresher course in chemistry, chemical technology, and pharmacy. Sir has published different research presentation and publications on his credit, that is total 43 papers, 29 national and 24 international. There is lot more. There are n number of incredible achievements of Devkate sir, but due to time constraint, I will stop here. Sir, we have indeed privilege to have you on our expert panel today, sir. Welcome you, sir. And I would like to request Dr. Devkate sir to start their session. Please, sir. Over to you, sir.
Okay, thank you very much, Chitra madam, for your kind word in introducing me. Uh, Actually, role of HPLC in Indian identification and purity checking of herbal drug is a topic of my discussion. I am very much thankful to principal and organizers of the KT Patil College of Pharmacy, Usmanabad, for giving me this opportunity to present or to discuss on this topic of the role of HPTLC. Everything is best. I'm not that much if it's efficient to teach you. Just I'm discussing my experience with you people when I'm doing this HPTLC in Amravati. We have this instrument at Amravati, and we have worked on this HPTLC instrument, and I found it very useful in herbal analysis. So I'm just making some daring to talk with you on this topic. So actually, the subject of this week faculty development program was very interesting for me. That is herbal medicine research and global health. So it is a very topic as per the today's era is concerned, because most of the safety is towards the herbal drugs, and we are shifting towards the herbal drugs. Actually, this is not today's era. If you think of the our history, you can find that in the Ayurveda, a lot many drugs are available for the treatment, and they are not explored to a great extent, and we need that. We should explore all those drugs, and such type of the program will definitely help all the participants to work in this area, because herbal technology, it is not the new technology. Our ancient books, charak and everything shows the use of herbal medicines in treatments. Even in this pandemic situations also, many herbal drugs has been used for treating such diseases, and that was shown by some researchers. So this is a very good part of your research. And for again isolation and characterizations, we need that we should have a specific method, and this role of HPTLC is an important part. Definitely it is an costlier equipment. Very few institute has this HPTLC equipment at their institute level. Even if you are not having this HPTLC at your institute level, CAMAG is supplying this HPLC, and it is marketed in India by Anchrome, and they have set up the lab in Mumbai for this HPTLC, and then they are offering the free training for the participants, as well as for the teachers of various colleges, institutions, academics, and they are helping the student by charging some cost to the student, because we know that we had to bring some marker compounds whenever we are doing the HPTLC analysis. We should have the marker compounds, and the cost of this marker compound is too much, because they are extracted from the plant. And in when we are doing work on this HPTLC, we find that we are not affordable to purchase all this marker compound by our own. So what we have tried to isolate marker compound at our level only. So in college, what we have done the preparative TLCs. If you know, if you identify that this is the component, if you take the spectroscopy of that component, and if you are able to identify it clearly, then what we can do, we can go for the preparative TLC. Okay, so just give a band on the TLC plate, simply what we are using in the laboratory. Okay, make a thick layer on the TLC plate. Then you develop this TLC plate, and when it develops to a certain distance, you scratch out the band, and you can extract that band with the suitable solvent, and whatever part will be there, that we can use as a marker compound. And that was my achievement when I was doing this HPTLC at Amravati, because every time it was not possible to purchase that marker compound. Okay, so if we are going to the Ancom laboratory, they are giving you this marker compound. Okay, they are definitely costing some part with this marker compound. Okay, so again they have certain this CAMAG is having a good website. If you have seen this CAMAG website, can I share the website on this screen? No, I'm not making any advertisement of CAMAG. I'm just showing you the potential of CAMAG in our research work, because many time what happen, we are not able to find out the exact mobile phases for the particular drug, and this mobile phase can be find out from the CAMAG. Subscribe their bulletin. Okay, so they are giving online bulletin on email. Those who staff who are involved in the herbal analysis, they can find it effective. I will share this HPTLC site of the CAMAC. Somewhat slow slightly. Okay.
Uh, so you can share the website, sir.
Can you share this? Can you see this website on my
Yes,
screen?
sir, yes. HPTLC. Yes, HPTLC. Yes, sir.
CAMAC.
Yes, 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 yes.
Okay. So this is only two companies are there who are supplying this HPTLC instrument. Actually, when it was developed, it was not included as a official method of analysis, because this HPTLC instrument, it is somewhat costlier. And again, um, it was not validated as such. In the previous era, it was not validated. So it was not included in official books. But after observing its potential, it has been included in all the official books, and HPTLC is used as a official method for the identification and purity findings. Okay, but only the suppliers, two suppliers are there. One is your CAMAC and one is your Chromeline. Chromeline is there. Densitometry, whatever Chromeline gives, densitometer, that much sophistication is not with the Chromeline, as the cost is also less. The Chromeline instruments are commonly utilized. See this website. Okay, so you can see product application, supports, education, new events, and about. In new events, you can find out their online training programs. Many online training programs are there in the new event area. Then this is the education. In educations, you will get this webinars, scientific publications, databases of CCBS, and that databases of CCBS they used to send by email also. I have this database for selection of the various mobile phases. Okay, so when we are doing separation of the secondary metabolites from the plant, and it was very difficult. So that is a cumulative CAMAC bibliography services. Okay, so it is a very important document that can be used. Okay, so you can browse any separation, full text search is there. Sorry. And you can browse out some products with this. I have just downloaded one for your references. I will show you how this CBSC CAMAC um, will help you selection. So it is a CBSC publications. Okay, so there cumulative CAMAC bibliographic services. So this is one document I had downloaded. You can see this document. Okay, so this is just introduction part of this CAMAC. So every topic is discussed in detail in this CAMAC services. So planar chromatography, we are talking about the planar chromatography. If you see the classification of chromatography, planar and columnar chromatography, this is not the presentation actually. Presentations will start, but I will show you just how this planar chromatography is there. Uh, Polycyclic aromatic hydrocarbons, not only for the herbal drugs. Okay, so it is used in all the fields. Okay, so this is for the hydrocarbon analysis. this it is used. Okay. I think internet is slow and you're not able to see. If you just see the index also, you can find out that it is very much poly polycyclic aromatic hydrocarbons road surfaces, then leucine and citrine in red yeast rice production. Okay, so see this simplified analysis of purity of the ginkgo go products, pharmacokinetics of barberine from Sardinia churna. Okay, then analysis of innovative plant extract. Okay, so 11 to 13. See, this is a very interesting chapter I have found in this. Okay, so analysis of innovative plant extract. Okay, I will show you this how it looks. Everything is given. What is chromatographic layer consisted of, then specific applications of this chromatography means fundamentals, result and discussion. Every part is given in very good manners. Okay, so this help you in identification. So what are the chromatographic layers which we can use? So this is the instrumentation. Actual part, page number 11, I will show you. Okay. So most of the time when we are asked to talk on this HPTLC, we used to refer this um, CAMAC website, because this is not only the TLC part. Okay, so um, the first part, automated TLC, is a HPTLC, and still this HPTLC is now combined with various more sophisticated instrument, and it has been converted to some hyponated techniques, and it was definitely used for the identification, characterizations also. Separation is only we was thinking for only the separation TLC, and now it is used for separation, identification, and even characterization, because it can be coupled with the mass spectrophotometers. It can be coupled with the capillary electrophoresis equipments. It can be coupled with n number of the equipments, and the team is involved in the searching of such type of the novel techniques. Okay, so that is a hyponated techniques. See, these are the certain books, review of the books on the TLC, fundamental theory and general principles of theory, general techniques of new apparatus techniques for sample preparations are there. Then special techniques they are given for the HPTLC. Okay, so this cumulative bibliography, I can share this document with you. Actually, they are sending these documents by email to their registered user only. And if it's possible, I will share this to your coordinator, and he can share all this part to you people. Pharmacokinetics of barberine and whatever our interest. Okay, so analysis of innovative plant extract. So this is the team who is working on this plant extract. HPTLC analysis of the fresh leaves extract was used to determine the optimal harvesting period. Okay, this is not only for identification, but even what and in which period, what is the environmental conditions which required for cultivation of the herbal product, and at what time the herbal product can be harvested. This part is also discussed here. Okay, so most important is what layer we should select. Okay, so HPTLC plate silica gel 60F 254 merc of 20 by 10 centimeters they are using for the separation and sample applications. The most thing is your mobile phase. Okay, so we stuck up in selection of the mobile phase, and definitely this will help. See this Messia officinalis leaves, which can be extracted by using and your simple extraction technique, and how they can be analyzed by using the HPTLC. So see, uh, for our practical purpose, optimization of the mobile phase, first we require that we should get optimized mobile phase, and then we are going for the separation and analysis. And if you refer these booklets, most of the job of optimization will be minimized. Okay, so our researchers spend most of time in selection of the mobile phase itself only, and it took a lot of time in selection only. And after that, then separation, characterization. So this is a lengthy period if you start from first step. Okay, so if you use these books, most of these mobile phases are available here, and the visualizing agent or derivatizing agents are available, which can be used for the research purpose. Like see here, it is a toluene ethylene ethyl format and formic acid in the ratio of 6:4:1 they are using. For quantitative ethyl acetate, water, acetic acid, for formic acid, that can also be used. Fingerprinting analysis is done in finding the migration distance of 70 सेंटीमीटर. ये सब क्या हो रहा है, इतना सब हमको मालूम नहीं है. आप जो बोल रहे हैं तो वो हमको कुछ important पार्ट मालूम नहीं है. Most of the things people say that it is very difficult to go and use this HPTLC. No friends, it is a very simple technique. If you are doing TLC in your laboratory na, then you start your job in your laboratory only, you finalize your research in the laboratory only, and for the final documentation purpose and everything, you take this to the Anchrom lab. Okay, so if you have prepared your own things before you reach to the Anchrom lab, then it will took a very small period of time, and very minimum cost will be affected, and will be required, and you will be able to complete your separations. Okay, so by using simple TLC plate also you can start your research, but for that you required a thorough literature survey. Certain books they are given in the Camac laboratory at Ancrom. I was there for the training program, and there was a huge library which was available with them. After completion means I was supposed to live there only. I don't go to my room. Okay, only for sleeping purpose I go to the room. I come back to their library, I just see whatever books, and even some books are available for purchase from them. I have purchased some book from for my college library, and I kept in Amravati. Actually, at that time I was working in Amravati, so those books are available in Amravati. If anyone are interested, you can see those book in the Amravati also. Egon Stahl book is there in everyone's library. Egon Stahl, HPTLC by Egon Stahl. Even Shettier sir is also given some books for the HPTLC. So you can refer those books at your library. But for detail, you can go for this. Okay, so this is a very documentation part, densitometry, which light you should use, what wavelength you should use, which plate you should use, result and discussion, everything is given. See, this is how beautifully these results are studied in case of HPTLC. These are the examples which we can see. This is the scanner. Okay, so scanner 4 is now available. So when I was using, it was scanner 3 at that time. Now scanner 4 is there. Linomat 5 I was using now. Various automated Linomat systems are available. Okay, so these are the Indian peoples who are working. This is not only for the foreign peoples. Okay, we peoples can also start working on this, and we can develop a good repo with this, even for the purification. Okay, not only for the herbal drugs. Okay, so I'm using this part for the impurity profiling. HPTLC used in the impurity profiling, because if you see the comparison, you will find its potential in your DT analysis also. Okay, so this I will share this document. You'll love to read this document. Okay, so you will say that we don't need anything more than this document. Okay, so it is a good document which is available. It is available at the website also. You can see this website. Okay, cumulative Then applications, many applications are there on this website. Application fields, application notes. Okay, directly you can go for the application notes. In the herbal analysis, I have found many notes on this website. Directly I contacted with the Kamak people also, and they are given me the good application notes on this analysis of quantitative control of lipid based formulations, then identification testing of sorbitol, then quantitative quantitation of carbohydrates in various materials. And this is free available. You can directly download this PDF. Okay, if you say this, you go and register, you can go and register with them. You can get all these documents free of cost. Okay, so this is the login which is given. If you enter your login, you will get this. Let me check whether I am able to do it now. Forgot password. This is the common problem because okay, let it be. I had to recover my password. Many time I used to forget my password. Okay, so now we will turn towards our topic actually of discussion for you people. Okay, so this is what I was supposed to give the idea of the software which is available there. So that definitely this will help you people. Okay, so what is the content of my today's talk? This was not as a part of today's talk only. Just I was doing. Uh, so I'm going to talk on the introduction principle of HPTLC, difference between TLC and HPTLC, difference between HPTLC and HPLC. Okay, so whatever difference will be there, we're going to talk on this. Okay, steps involved in HPTLC: material used for plates, mobile phases, sample preparation, application, and visualization. So this part we are going to discuss in today's presentation. Can you see everything?
Yes, sir. Yes, we can.
May I stop my video? Why this is so delayed? If I stop my video, it work fast
No, sir. No, not the problem of video, sir. It is, I think, network.
because it's taking too much time to complete. ओके वैसे भी मेरे
Okay,
सर पे
okay,
कुछ नहीं है इतना मेरा मुंह तो वैसे ही वहां पे लटका हुआ है साइड में इट इज नॉट
sir.
नेसेसरी टू शो माय वीडियो definitely it will increase the speed of our presentation. Okay, so
Okay,
I'm
sir.
going to discuss
No
on
problem.
this part: introduction principle of HPTLC, difference between TLC and HPTLC, difference between HPTLC and HPLC, steps involved in HPTLC, material used for the plate constructions, mobile phases which are used for the HPTLC separations, sample preparation, application and visualization in case of the HPTLC, application in herbal drug analysis with certain examples. Lot many examples I found on the websites, and I can discuss all these examples with you people. So, what is a high performance thin layer chromatography? Okay, before we have certain chat with the participants because it becoming a very monotonous. Let them
It's
participate.
not
Can they?
interactive sessions. Actually, this is one-way interaction, sir.
Only one way
Whatever the
communications.
questions,
Okay,
yes,
okay.
sir.
Let
Whatever the
it be.
questions will be there, they will
Okay,
add in
let
chat
be.
box.
If they can, they can ask the questions in the chat box
Yes,
also.
yes, sir.
So
Yes, sir.
chromatography. Before we proceed, we can just think of the chromatography. What is the chromatography? Why it is used? Chromatography, that common definitions, what I am going to explain, it is a physical method of separation involving differential distribution between the two phases. One phase is percolated through the another. The former is known as a mobile phase and the latter is known as a stationary phase. This is a very common definitions what we are teaching to the students. But how much of we people actually understood these definitions? These definitions given every principle of separation. If we ask our student for the classification, they say TLC, paper chromatography, gas chromatography. This is not the classification. Okay, we should tell them that chromatography is not classified in this way. Either we can classify it on the depth of separation, that is planar chromatography or columnar chromatography, or we can say that on the mobile phase use, we can classify the chromatography as a liquid chromatography, gas chromatography, or superfluid chromatography. This is the second method of classification. And the third last method, what we are commonly using, is depending upon the mechanism of separations. What is the differential distribution mechanism? Either that involves the separation by adsorption, that is adsorption chromatography. We call it as a adsorption, or solid stationary phases are used. Second is your partition chromatography, where liquid stationary phases are used. Or we can use the ion exchange resin, it goes as the ion exchange chromatography. We use the gels, that is the gel permeation chromatography, okay, or molecular sievings. Then if you are using the ionic species, that is ion chromatography. Means all this can be coupled in the HPLC itself only. Ion exchange columns are there in case of HPLC I'm talking now. And most of the things has been modified for the HPTLC also. So it is a high performance thin layer chromatography. Only the difference in HPLC and HPTLC. HPLC also called as a high pressure, and here we are not calling it as a high pressure. It is only the performance. And why the performance is there? That point we have to look out. That is, HPTLC is a sophisticated form of TLC. Okay, what TLC we are using? It is only the sophisticated form which provides superior separation efficiencies. The advantages of automation, scanning, full optimization, selective detection principle, minimum sample preparation, hyponation, means by combining this HPTLC technique with all the modern spectroscopic technique, that is hyponations, and so on, enable it to the powerful analytical tool for chromatographic information of complex mixture of pharmaceuticals. Okay, so we are mostly utilizing for all pharmaceuticals, not only for the herbals. Okay, natural products, what we are talking. Okay, natural product, not only the herbals, every natural products, even the minerals that can be obtained, petrochemicals that can be obtained. So it can be used for all natural products. Then clinical samples. Okay, it has got more applications in clinical area also. Okay, so clinical samples can be analyzed. Foodstuffs in food industry, it also has great applications in identifications. Then and so on. लॉट मेनी टेक जित जहां जहां हम लोग HPLC इस्तेमाल कर रहे हैं, वहां वहां अपने को we can use this HPTLC there also. The HPTLC concept includes validation methods for quantitative and qualitative analysis and fulfills all qualitative requirements for use in fully regulated environment. Actually, when it was started, it was not approved as a official method of analysis. Okay, because it was not involving the validation parameter. And now they have developed this validation technology also, and if the validation technology are there, they can be included as in the official monographs. Okay, if you see old official books, they does not have this HPTLC as a monograph component. Now it was included in their monographs also. Okay, so because they are having this regulatory environment requirements. Okay, like your 21 CFR. Okay, what is 21 CFR? That is Code of Federal Regulations. According to that, we cannot change anything during the process. Okay, it requires the prior approval for any minute changes in your method also. Even if it is a analytical methods, you cannot change by your mind. आज नहीं आया अपने प्रोडक्ट, तो हम लोग उसको थोड़ा सा कुछ चेंज कर लेंगे, नो. So they required a complete legal procedures for changing all these things. So, introducing chromatography, I already discussed with you regarding the chromatography. It is a physical method of separation involving differential distribution between the two phases. One phase is percolated through the another. The former is known as a mobile phase, and latter is known as a stationary phase. So, this is not the definitions given in book. Okay, IUPC definitions or some other definitions you see, it has given everything. It is chromatography is a physical method of separation. Okay, certain physical parameters can be utilized for separation of the material, and the basic separation is based on the differential distributions. Okay, so either there may be the partition, there may be the adsorption, there may be the ion exchange, there may be the affinity chromatography, there may be certain ionic strength, or any methods can be used. And that basic principle is your differential distribution, involving two phases, stationary phase and mobile phase. Every time there is two phases, one is known as the stationary phase and mobile phase. So if you look out the history of your HPTLC, in that terms only I will talk. I will not start with the 1904, where Twist has developed this column chromatography for separation of the plant pigments on the chalk column. But I will just only start from 1960. In mid 1960, the first instruments were developed for scanning densitometry. Okay, densitometry was the first equipment developed. Densitometers, they are determining optical activity, optical density, we can call it as. Absorption term was arrived afterward. Okay, so they were used to determine the optical density of the material on some slides, and that was the densitometry. If you see our old equipments available, such type of the densitometer was available with us also. HPTLC तो बहुत बाद में आया, लेकिन जब हम लोग पुराने कॉलेजेस के डेड स्टॉक देखेंगे तो उसके अंदर ये ऐसा एक था डेंसिटोमीटर. Okay, so that densitometer was used to measure the densities on the TLC plate. Okay, so they uses the absorbance or fluorescence measurement in reflectance or transmittance mode. अभी तक तो हम स्पेक्ट्रोस्कोपी की तरफ सिर्फ ट्रांसमिटेंस मेजरमेंट ही देख रहे थे. Okay, so reflectance phenomena we were using in IR also. Diffuse reflectance assemblies, ATR, attenuated total reflectance assemblies. Okay, if you see the principle of this diffuse reflectance and attenuated total reflectance, the same principle is there in the HPTLC also. Okay, there is also we are measuring the samples on the surface. We are using this IR for the quantitative estimations. Then why can't we use this HPTLC for quantitative estimations? That was thought of by the peoples, and they use these principles for the separations. The commercial production of HPTLC plates in the mid 1970 was started with the Merck. Okay, so Merck company was first to develop this in 1973. Herbal introduced first nano TLC plates. So what is the difference between the TLC and HPTLC? So that will tell you how the history has been evolved for the development of this HPTLC. What we are thinking, why we are starting with history. Okay, so there lies the basic things. Whatever mechanism of separation was there, you will get that basic idea from their history. So we should read the history before we directly jump out to the directly instrumentation and handling of the machine. How they come to today's instrumentation. Okay, so that is the densitometry, and densitometry again was used in many things, many areas. The basic separations can be achieved in short migration distances. That is the difference in the TLC and HPTLC. The analysis type is greatly reduced by HPTLC. Okay. Um, o- Only one quantitative techniques we have this HPLC. Okay, so you if you think of the limitations of HPTLC, then you will definitely prefer HPLC. Then you will definitely prefer HPTLC for your analysis. We'll discuss all these limitations here. Okay, so, smaller diameter of sample spot applied HPTLC allows the analysis of 20 to 30 samples on single plate. Okay, 36 samples we can apply on to a plate of 20 centimeter length. Okay, so that is a very effective methods. So if you think what is the difference between TLC and HPTLC, This table will summarize the differences between the TLC and HPTLC. Okay, the main particle size, why high performance? If you think it's mechanism, of the performance, that is due to the particle size only. This particle size you have seen in the HPLC, 5 micrometer. Same particle size we are using in TLC plates. If you use your simple TLC, it is not minimum 20 micrometers. 20 micrometer and above we are using in TLCs. Okay, as you reduce the particle size, it will increases the surface area, and it will gives you the good separation efficiencies. Therefore, the term high performance. The main performing characteristic is this, not only the pressure. Okay, in HPLC, we are finding the pressure as a performance characteristic. No, the performance characteristic is the particles, and due to the reduction in particles, there is a resistance to flow of the liquid through the column, and that requires the high pressure. Therefore, pressure is increased, but there is no penetration through the column, and that is the beauty of this TLC. Only the surface movement of the liquid is there, and even if you reduces the particle size, it will not affect the flow properties. Okay, so that increases the high performance, and therefore HPTLC, this only the more important thing in HPTLC. Again, the thickness layer. In case of the HPTLC, we are having the thickness of 350 micrometer. In case of HPTLC, it is only the 100 micrometer. Pre-coated plates are available, okay, which are maintaining the even thickness on the surface. In the laboratory, when we used to prepare this TLC plate, still we are using. Okay, so we use either dip methods or pour methods. Okay, if you go and see the history, the applicators were developed for application of this layer on the surface, where we can adjust the length from 1 mm to 2 mm in millimeters. Okay, so either we can go for the preparative TLCs or simple TLCs by using this applicator, but they are having the lot many disadvantages. Okay, we required a large quantity of these slurries when you are using the applicators. But in case of the HPTLC, only the pre-coated plates are available, and they are not too much costly. Okay, so that is the main thing. The thickness of the layer is also reduced, and the migration distance again. We have to migrate it for long distance in case of simple TLC. Even if you have developed the TLC of your sample in your laboratory and you take it to the HPTLC laboratory, so you can observe all these differences, that they are developing the chromatogram only to 3 to 6 centimeter only, and in your TLC we have to develop the chromatogram up to 10 to 15 centimeter for better separations. Otherwise, you will not get the separated components. Okay, typical separation times, it is somewhat time consuming in TLC. That is, we are having to go for 20 to 200 minutes period of time. In case of this, even it starts from 3 minutes, because only 3 to 6 centimeters we have to go. See the ratio, 6 as to 20. Okay, how much ratio, that will decide how much time you will take. Okay, so generally 20 minutes is more sufficient for all separation process in HPTLC. Okay, so number of sample per plate in TLC, 2 centimeter distance we are keeping between the two drops, two spots. Here we can go up to 36, more than 36 spot in a single plate. Definitely the plate size is important. Okay, so if you are using small plate, you cannot say that Devkada sir has this 36 spot, I will put 10 centimeter slide and give 36 plot. No, 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 no. You have to go for the length. Ratio is also there. Okay, if your spot is 6 mm in diameter, okay, so sufficient distance should be there also. Otherwise, mixing of spot will be there. And the most important, the second importance of your HPTLC is your solid support. What sort of solid support we are using? In case of your simple TLC, we are using silica gel with the normal phase chromatography, alumina, Kisalgar. Now you should know what is normal phase chromatography and what is reverse phase chromatography. I used to have a discussion with student and getting very much difficulty when we are talking. Okay, so in the normal phase chromatography, what is the nature of your mobile phase and what is the nature of your stationary phase? Stationary phase are generally polar, and your mobile phase are generally non-polar. But in case of the reverse phase, we have this mobile phase as a polar and stationary phase as a non-polar, and that can be achieved by modification of the silica gel. Okay, we can modify the silica gel. Why we find HPLC is more efficient? Because we are using reverse phase chromatography there. Here also we can go with the HPTLC reverse phase chromatography. That is the main advantage of HPTLC. In simple TLC, we cannot go for the reverse phase chromatography. That is normal phase chromatography also. Okay. So in case of the HPTLC, we can go for the reverse phase chromatography. Silica gel which is modified for the stationary phase, means there is a bonding of certain functional groups with this stationary phase, and that will help for the separation. Okay, means C18 type of the column if we are using there, we can have the C18 plates here in the case of HPTLC. We can have the C8 plate, we can have the nitrile plates. Various stationary phase can be used in case of the HPTLC. Okay, so solid supports which are modified can be used as a reverse phase in HPTLC. Then mobile phase requirement, that is again see HPTLC. We require only 7 to 8 ml of the mobile phase, because whatever mobile tanks we are using, okay, so they are modified to use for the HPTLC. Okay, and the efficiency, as all this will definitely increase the efficiency of your HPTLC plates. Now, if you see, main difference of HPLC and HPTLC. Now, see, even we can think that HPTLC is more preferable than HPLC. Okay, it was included in all USP monographs, but still you can find it has more applications than HPLC. How? We can see here simultaneous processing of samples and standard under the similar conditions, leading to better analytical precisions and accuracy of analysis, less need of the internal standard. Okay, if you have seen the HPLC procedures, we required to inject the internal standards. Okay, we had to go for the system suitability test in case of the HPLC, whether my systems will produce the reproducible results or not, because the injection of sample is different. For every sample we have to give the separate injections. If it is simultaneous, the single injection will do, but for quantitative we have to give only different injections for 10 ppm, 2 ppm, 4 ppm, 6 ppm, 8 ppm, 10 ppm. In HPTLC we can have the spots on the same slide of all the concentration together. So simultaneous is only possible with HPTLC, not on the HPLC also. Okay, so that is the bigger advantages of your HPTLC over the HPLC. यहाँ पे HPLC में एक एक सैंपल आगे आगे जाएगा और बाद में सेपरेट हो जाएगा. HPTLC में सब साथ में सेपरेट हो रहे. They are having the equal environment while separating and giving the more efficient separations. So it has a flexibility of extreme flexibility for sampling of various samples. Okay, here only like dissolves like. Polarity and solubility is important in case of HPLC. Equipments are very costly. If your sample is not soluble, then it will get adhere into the column. It will block the columns here to throw the columns of 40,000 rupees. यहाँ पे कुछ नहीं है, एक प्लेट में अगर वो आप चिपक के भी बैठा तो वी कैन थ्रो द प्लेट बिकॉज द कॉस्ट इज वेरी स्मॉल and therefore it is more flexible than your HPLC. Then operation skills, less skill is needed for operating HPTLC once you have given the proper trainings. Every times you don't require a technical knowledge about this. एक बार आपको अगर ट्रेनिंग दे दिया HPTLC इस्तेमाल करने का, you can use it for the different samples. Okay, you can think, you can explore your thoughts while doing it without more skilled requirement as the HPLC, because it is simple to learn and operate. High sample throughput of the similar or different nature of samples means lower analysis times and less cost per analysis. ओके, अलग अलग अगर उसमें अपने पास में सब्सटेंसेस है, ओके, पॉली हर्बल फॉर्मुलेशंस का बहुत चयन निकल गया अभी आज तक तो हम देख रहे हैं। जैसे ही वो पाइल्स का टैबलेट चालू हुआ था ना, सबसे पहले पॉली हर्बल फॉर्मुलेशंस में पाइल्स टैबलेट आ गया, 24 ड्रग्स उसके अंदर डालते, हर्बल ड्रग्स, और उसका 24 ड्रग का एनालिसिस करते बैठो तो कितना डिफिकल्ट है बाकी सिस्टम से। लेकिन अगर हम लोग हर्बल अगर लेते, पाइल्स टैबलेट, पाइलॉन, पाइलॉन जो टैबलेट था, मैंने पहले सबसे पहले उसके ऊपर काम किया था, पाइलॉन, एंड आई वाज वेरी मच tired in finding the mobile phases for all components which are there in the pylon tablet, 24 components. It makes me very difficult for choosing the solvent mobile phases, but that was made very easy by using this camag bibliography. बहुत सा टाइम उसके अंदर गया था हमारा ढूंढने में। ओके, सो हाई सैंपल थ्रूपुट इज द मेन एडवांटेज ऑफ HPTLC. ओके, कॉस्ट मैंने जैसे बोला कि एक बार हम लोग HPTLC का कॉलम जो यूज करते हैं, HPTLC की प्लेट, तो उसका कॉस्ट ज्यादा से ज्यादा, रिवर्स फेस भी है तो 150-150 रुपए तक आ जाएगा। हमारे वक्त में उतना था, अभी तो पता नहीं मुझे, क्योंकि मैं जब से अमरावती से ट्रांसफर होके आया तो आई डोंट हैव दिस HPTLC टू वर्क विथ मी, बट स्टिल आई फाइंड दैट इट कॉस्ट इज टू मच लेस एज कम्पेयर टू योर HPLC कॉलम्स, because HPLC column starts from 40,000 rupees, and the cost is not of the single column, it has a cost inclusive of your guard column also. Okay, so guard column cost is there, then simple column cost is there, so total cost of your separation is more. And even if you are injecting the plant extract, what we used to think that इतना तो सब हमने कर लिया, इतना कॉस्टली इक्विपमेंट ले लिया, we will inject the sample and let the machine separate out all the things. No friends, machine will not separate anything. It will get clogged if you are directly injecting the extract. Okay, many M.Pharm students thought that we will prepare the extract and directly inject into the HPLC. That much costly equipment is there. No, your extract will block all the HPLC columns, and only you have to throw the columns, purchase a new one. You required further fractionations on the simple columns or TLC, and then whatever fractionated components are there, that you have to inject, not the extract. Okay, so this cost of the column is too much in case of your HPLC. Then second is sample preparation. एक तो भाग जाता आगे, नहीं तो एकदम पीछे चले जाता। हाँ, सैंपल प्रिपरेशन सिंपल है, इसके अंदर कुछ नहीं है। उसके अंदर देखो, आप देखते होंगे कि वी हैव टू फिल्टर दैट सैंपल एंड मोबाइल फेजेस थ्रू द मेम्ब्रेन फिल्टर्स और द थ्रू द नायलॉन फिल्टर्स, 0.42 plus minus 0.02, 0.22 plus minus 0.02 to take out all your particle matter from the solvent, even from your sample also. Okay, because we are very much afraid of clogging of this column, and that is not there with your simple HPTLC, because we are just spotting on the surface. The solvent system, solvent needs to be prior treatment like filtrations and degassing. In case of your HPLC, here we don't require any sort of such purity of the samples, solvents. ओके, HPLC ग्रेड सॉल्वेंट इस्तेमाल करने की जरूरत नहीं है वहां पे। वी आर जस्ट यूजिंग द HPLC ग्रेड सॉल्वेंट्स, हियर सिंपल सॉल्वेंट्स कैन डू. Okay, sample applications, variable volumes can be applied. जैसे मैंने बोल रहा था वहां पे कैसा है कि अगर आपका लूप इंजेक्टर है तो फिक्स वॉल्यूम रहेगा 20 माइक्रोलीटर लूप, what machine you have, accordingly that you work in your laboratories. Okay, so there is a fixed quantity you have to inject, but here we can have a spot of 6 with varying quantity of the sample in it. उसका स्पॉट का जो ये जो है बैंड का, तो उसका डायमीटर और उसका लेंथ कुछ चेंज नहीं होने वाला है, लेकिन उसके अंदर का सॉल्यूशन का लेंथ वी कैन चेंज. Okay, so we can apply the spots or bands. Here we are doing the injection by the syringes, manual as well as automatic injectors we are using here. We can also have the manual applicators, or we can have the automated application systems developed for HPTLC. Okay, so we can have the solvent grades, HPLC grades. So what steps are involved in HPTLC? That selection of chromatographic layer. Okay, here we have to decide what chromatographic layer we should go. That you should know the nature of sample which you are separating. Accordingly, that we can select the chromatographic layer. Then second is the layer pre-washing. Okay, तो वहां पे जैसे वॉशिंग करते हैं हम लोग कॉलम का, तो वहां पे यहाँ पे भी प्री वॉशिंग है. ओके, टीएलसी में हम लोग प्री वॉशिंग नहीं करते हैं, बिकॉज वी यूज टू प्रिपेयर द फ्रेश टीएलसी प्लेट्स एंड यूज. यहाँ पे वो प्री कोटेड प्लेट्स आते हैं, तो जब भी वो रखे रखे रहते लेबोरेटरी में, वी रिक्वायर्ड दैट दे मे एब्जॉर्ब, as they are the absorbent, they are having the nature of absorbing all the atmospheric components on their surface, and to remove them we require the pre-washing step. So the pre-washing step is there. Then sample and standard preparations, we had to prepare the sample and standard for comparison purpose. Then we have to apply these samples by using the suitable methods. Then chromatographic developments. Okay, so whatever chromatographic developments we are using. Then a detection of spot. Okay, so these steps are there in your laboratory also. Only the layer pre-wash is not there. Okay, in simple TLC we are doing all these things. Then scanning and documentation of chromatographic plate, and that is the regulatory part which is helping this HPTLC to be a validated techniques. Okay, scanning and documentation. Scanning will give you the quantitative results, and documentations will help in your regulatory aspects. Okay, so that is only the difference between your TLC HPTLC working. ओके, हम लोग तो वैसे भी टीएलसी का फोटो लेके उसको बोलते हैं कि हमने डॉक्यूमेंटेशन कंप्लीट कर लिया है, लेकिन वो डॉक्यूमेंटेशन है, इमेज एनालिसिस इज नॉट देयर with your simple photographs. Here we are doing the image analysis also. Okay, so that part is there in the HPTLC image analysis. So plate materials, what plate, एक बार इसको एक ही बार दबाना पड़ता है, थोड़ा स्लो रिस्पॉन्स. ओके सर, कितना टाइम दिया है मैडम मेरे को?
Sir, up to 1 o'clock, you can continue, sir.
वी हैव वन मोर ले आवर ना.
Yes, yes,
ओके
sir.
ओके, नहीं तो मेरा बहुत स्लो चल रहा होगा, तो नहीं तो मुझे फास्ट टाइम करना
Yes,
पड़ेगा, नहीं तो वो प्रोसेस.
yes. 1 बजे
Okay,
तक.
I will
Yes,
continue up to 1 hour, 1 o'clock.
yes,
Okay,
yes,
so we also had to start discussing. So plates and
yes.
materials, the support materials we are using, generally the glass in laboratory, we are using this glass for the TLC purpose. But for HPTLC, we are having the aluminum sheets or polystyrene sheets. Polyester sheets are also available today. Okay, so what is the basic advantages of polyester sheets, that will look out. Then what is the advantages and disadvantages of aluminum sheet, that also we will discuss. But we are more discussing on the sorbent use. Okay, fluorescent silicas. Many time our sample is not fluorescent in nature, but what we can use, we can have the background of the fluorescent nature. Okay, automatically the sample will get visualized even if it is not having the fluorescence. Okay, so that is a fluorescent silica gels we can use. So that is a 60. Then aluminum oxides we can use as a adsorbent layers, cellulose, silica gel, chemically modified, that what I am discussing, C8, C18. So we have these supporting materials like glass. Okay, so what is the advantages? Resistance to heat and chemicals, offers superior flat surfaces of work, but it also has certain disadvantages, that is fragility. Okay, glass are very fragile. So my friends, if you are working in teaching field, we can Find out all the broken glasses of your college. खिड़की के जो कांच रहते वैसे भी मैं उठा के लेके गया था। सिर्फ वो ग्राउंड ग्लास नहीं होना चाहिए, प्लेन ग्लास होना चाहिए। या फिर फ्रेम मेकर्स के पास जाके, इफ यू आर डूइंग टीएलसी, we required minimum apparatus, and you just go to the glass maker, frame maker, उसके टट्टे फुटे ग्लास जो रहते हैं उसको बोलना ऐसे रेक्टेंगल रेक्टेंगल में कट करके दे दे मुझे। Okay, you can develop your setups in the laboratory easily using the class for your first step of development. Okay, सब लोग वो छोटा सा स्लाइड लेके करते हैं। स्लाइड में नहीं चलता है, एक स्पॉट देने का है तो स्लाइड यूज कर सकते हैं, एक या दो स्पॉट में। अगर अपने को बिगर स्पॉट देने के, एन नंबर ऑफ स्पॉट देने के, तो अपने को उसका साइज बढ़ाना पड़ेगा। साइज बढ़ाने के लिए simple way to go to the frame makers, and whatever broken glasses are there, you find out the glass of 10 centimeter to 15 centimeter areas. Just excuse me, I will have to. Sorry for interruption. I had to drink some water. Okay, so we are having a simple method to make these plates, that are glass plates, which are used in your day-to-day life. So that can be used, but in case of HPTLC, that cannot be used because they are fragile. Okay, and relatively high weights. But if you are thinking of aluminum sheets and polyester sheets, Merck plates are available. Other companies are also providing with 0.2 mm thickness surfaces. Okay, or 0.1 mm thickness surfaces, aluminum can be used, but polysters are using with the 0.2 mm thickness. They are having the more economical, unbreakable, less packing materials are required. Many plates can be packed in small area. Okay, carrying is very simple. Okay, so we can take out these plates, pre-coated plates, to any area with you. So carry but what is the basic problem, that if you are doing activations with the polyester plates, we cannot use 121 degree centigrade for activation, because this polyester may get melted. Okay, they may get molten at the higher temperatures. So activations with the polyester plates is not good, but aluminum sheets can be used. Okay, uh, they are unstable as such polyesters. They have the ability to react with certain organic solvents, and maybe not used in those cases. But if you are using some polymers which are resistant to all these solvents, they can be very well used. So preferred is your aluminum sheets. So we are having these aluminum sheets in the laboratory. So it has increased temperature resistance. We can heat at 120 degrees centigrade for activation, and they are very easy to handle. Even we can cut with the series scissors. Okay, in preparation of plate, I will discuss this matter with you, that when we are cutting, actually when you'll work in laboratory na, and when you'll actually handle those plates, you will come to know how to cut these plates, and how to handle these plates. But it also has certain disadvantages. Eluents containing high concentration of the mineral acids. Okay, if your mineral acids are there, if your mobile phase contains HCl, H2SO4 like acids, okay, so they will corrode this TLC supporting plates of aluminum, and they cannot be used. Okay, so if even the ammonia is there, that can also has the reactivity with the aluminum. Okay, so ammonia can attack the chemically on the aluminum. So we have to see what mobile phase is there. Then you have to select this material of the TLC plate. So what stationary phases are available? So just this is the support for this stationary phase. Simple, if you are doing the TLC, we can have the silica gel G, silica gel GF, and silica gel H. Same plates are also available with the your HPTLC. So silica gel without binder is known as a silica gel H. Silica gel G, that is gypsum with binder. Okay, so that can be used in your normal laboratory practices. Okay, here also the silica gel G is there with HPTLC, but generally we are not using G, we are using GF. That is fluorescent materials are applied on the surfaces that can be used as a background. Okay, silica gel GF. We cannot use silica gel GF in the laboratory for your simple TLC plates. Okay, aluminum oxides with or without binders can be used. Then cellulose powder with or without binders can be used. So generally in we are having silica gel GF. G stand for gypsum, F stand for fluorescence indicators. Like zinc selenate is added in the silica gel, which is giving the fluorescent nature. So we can have the silica gel PF, that is for preparative when we are doing in the laboratory. And you don't want to interact the material because we are separating it as your marker compound. Okay, so we are not doing any derivatization, and even your sample is not fluorescent. In that cases, you bring this silica gel PF, you make a thick layer of this silica gel PF on your surface of the glass and use it for the preparative purpose. Whatever spots are there, you scratch out the spots area. You just see the spots in under the UV light, and in UV light only you scratch out in the small petri plates or some glass watch plates, and then you extract that silica layer for your active markers, and you can use by yourself. Okay, so that is the most common thing what we are doing in the laboratory. Silica gel HF254. 254 stands for the wavelength at which it will show you the fluorescence. It contains binders and fluorescence indicators. So these are certain plates for the normal phase chromatography. In the reverse phase chromatography, we have certain reverse phase material which are bounded with the silica gel. Okay, unmodified forms of this silica gel is used. The analysis of 80% drugs is possible with this silica gel 60F. Okay, then aluminum oxide. So either basic substances, alkalides, and steroids can be used. So it is available in three forms: acidic alumina, basic alumina, or neutral alumina. Okay, so acidic alumina for separation of acidic substances, basic alumina for separation of the basic substances, and neutral alumina for separation of alkalides and steroids that can be used. So aluminum oxide is nothing but the alumina, which is used as a supporting material. Cellulose, macrocrystalline cellulose are there. Heavy cellulose or light cellulose are there. Means number of glucopyranate unit which are attached with this cellulose that can be used, suitable for amino acids, peptides, sugars, and other label compounds which cannot be chromatographed on the active layers of silica gels that can be used. Then modified silica gels are there. Okay, so C18 columns, C8 columns, or nitrile columns are there. Similarly, plates are also there. If you are modifying with ammonia amino groups, that is also available. Then we can modify it with CN. So COH, phenols, nucleotides, pharmaceutical, and preservatives can be separated on these modified silica gels easily. So plate size, what plate size we should select? So either we can go for, generally they are available in 20 by 20 centimeter plate size, but in looking towards the economy of our working, we used to cut this plate in the 5 to 5 centimeter, even 2 centimeter to 5 centimeters, but they cannot be fixed onto this HPTLC platform. So at least you should have 5 to 10 centimeter plates, 5 into 10 centimeter plate size for the HPTLC platforms. Okay, mostly we are using 10 to 20 centimeter plates or 5 to 5 centimeter plates for the purpose. Okay, so we can cut these plates, so you have to be very careful when cutting these plates. Okay, so these plates can be cut by using your scissor, so you should have the sharp scissor. Otherwise, when you are cutting, the layer will get peeled out. Or we can have the knife cutters for this purpose. And when we are doing cutting, we don't have to cut from the coated area. From the back side of this plate, we have to mark um, the pencil lines, and we have to cut with the cutters which are available. Okay, if you are using scissors, scissor work will work from both areas, and it will may produce the peeling out effect on this plate, or plate may get damage on the surfaces where you are actually doing the cutting. So instead of cutting with the scissor, you go and prefer the knife for cutting purpose. Paper cutters, sharp paper cutters we can use, and that is to be used from the back end of your HPTLC plates, not from the surfaces. Okay, HP जे cut केलेलं आहे, जसं when we are cutting the tomato. Okay, so you cut the tomato from half, and then you make the small pieces. नहीं नहीं तो हम पीछे से अगर कट करने को जाएंगे तो चिपक जाएगा, उसमें से पानी निकल जाएगा, वैसे ही इधर भी. Okay, so what size of the paper sheet is to be selected, that depend upon you, what samples, how many samples you are applying. Okay, so pre-washings of pre-coated plates. Why we required the pre-washing? Because whenever we are purchasing these plates, we are keeping with our cupboards, okay, which are there in the laboratory. And in laboratory, lot many environment is there. So this environment can absorb all the gases on the surface, because these are the absorbents, and their ability to absorb all the waters which is there, all the vapors which is there in the environment of your laboratory. ओके, पहले लाया था तब सफेद रहता है, और कुछ दिनों के बाद वो येलो हो जाता है, ब्राउन हो जाता है सरफेस. We find that it is now it is not useful. No, you go for the pre-washing. Even if it is a fresh plate, we don't require to get it pre-washed. But if you are kept for even one day, we require to get it pre-washed for the gases which are there. Again, when we are drying or activating or keeping these plates in the laboratory, you make one habit that you keep these plates in two glass plate surfaces. Okay, तो दो बड़े बड़े ग्लास प्लेट लेके आओ, उसके अंदर ये शीट रखो. नहीं तो क्या हो जाएगा कि टेम्परेचर के कारण और चेंजेस इन टेम्परेचर में कॉज दिस थर्मल कॉन्ट्रैक्शन ऑफ दिस एल्यूमिनियम, एंड वो ऐसा सिकुड़ जाएंगे. जैसे ही वो सिकुड़ जाते तो उसके ऊपर वाला जो लेयर है, दैट विल गेट पील्ड आउट, that has a tendency to peel out. So instead of keeping as such in normal way, you keep it into the sandwich between the two glass plates and keep it in your laboratory. Okay, so absorption will also be less, and they will not change their shape. नहीं तो वो टेढ़े मेढ़े हो जाते. Okay, so keeping, storing of plate is important, and pre-washing is important. So there are the various pre-washing methods. Ascending, सिंपल क्रोमेटोग्राफी जैसा कर रहे हैं वैसे ही सॉल्वेंट के साथ में उसको असेंडिंग क्रोमेटोग्राफी के लिए रख दो. और जो ऊपर वाला लेयर है, व्हाट वी यूज टू डू, उसको एक तो पूरा वॉश कर लो, दूसरे साइड से निकल जाए जब तक मोबाइल फेस तब तक, या फिर ऊपर जब सब जमा हो जाता है तो ऊपर का 2 सेंटीमीटर या 1 सेंटीमीटर का सरफेस जो है वो काट दो, जहां पे वो पूरा जो डर्ट है उसके अंदर का, तो वो कैरी करके आगे लेके जा रहा है. एंड वी कैन flow, allow to flow up to that level. You cut that area, remove from your plate, and you can use it as a fresh plate. Okay, another method is the dipping. You can dip this plate into the certain washing liquids, or we can have a continuous process of washings when you are using the automated systems. Okay, so we are not using actually the automated process. Mostly we are using the manual one only. So what are the solvents which can be used? Generally, we are using the methanol as a pre-washing solvents. That is sufficient for pre-washing of your plate if you are using in your laboratory. Okay, methanol, N-hexane mixtures we are using. Okay, N-hexane also we use, but mostly we are not using the non-polar materials on the surface. We can have to use certain polar. So we can have the chloroform methanol admixtures of 1:1 ratio. That is also good. Okay, either you use the plain methanol if you are frightened about the chloroform nature. Then if chloroform nature यानी कि वो उसके absorption. Okay, chloroform also has the absorption abilities. Okay, so अगर drying करते हो, activation करते हो तो chloroform will remove. Okay, so it will not interfere, methanol will get removed. So we can have the mixture of chloroform and methanol 1:1, or we can have the chloroform, methanol, or ammonia for some alkaline substances if it is there. Then we required to remove with the use of ammonia. We have to to change the pH. Okay, if the substances are not getting removed by simple methanol, we can have to add ammonia in only one part. 90 parts should of the chloroform, 10 parts should be of the methanol, and one part form of the ammonia. Then methylene chloride and methanol admixtures can be used, or simple ammonia solutions if it is more adhere with certain. और हम तो क्या करते थे, when we are using these plates we used to wash it again and again. जब तक फाइनल रिपोर्ट नहीं लेने का है तब तक उसको वही वही प्लेट हम लोग यूज करते थे, प्री वॉश करके. Okay, because जैसे finance is our first important things when we are working in the laboratory. If we do not have the sufficient finance, we used to find out certain easy ways. तो उसके लिए हम लोग अगर बहुत ही ज्यादा उसमें कंटामिनेशन, तो वी कैन गो फॉर द अमोनिया सॉल्यूशन 1%. Okay, after washing for the sufficient time for removal of the washed liquids, you had to dry the plates into certain desiccated oven, and never store the plate in the normal atmosphere. Either you store it into the desiccators, okay, or into the um, fume hoods मध्ये सुद्धा नाही. Okay, you should have to store in the desiccators only. Avoid hot cold air drying, otherwise it will cause the peeling out effect. Okay, so we had to do the activation of pre-coated plates. Fresh open box of HPTLC plates do not require activations. Other activated by placing it at a 110 degrees centigrade or 120 degrees centigrade for 30 minutes. Dry in a vertical positions when we are keeping in the oven. You have to be very much um, cautious. Okay, नहीं तो ओवन का ही सब कचरा उसको लग जाएगा. So it should be kept in vertical positions. Some racks we can prepare, or some holders we can prepare to keep these plates into the oven. अभी वो होल्डर का भी साइज अलग अलग होना चाहिए. ओके, क्योंकि अपने प्लेट तो अलग अलग साइज के रहते हैं. So mobile phases, chemical properties of analyst and sorbent layer factors should be considered while selection of the mobile phases. Okay, like dissolves like. So if you are having the polar substances to be separated, so you should use the polar mobile phases. And for that polar mobile phases, you should select the proper solvents. Your mobile phase may be a single phase systems, or it may be a biphasic systems. Generally, we are having a monophasic system only. It may be comprising of the single mobile phase, or it may be the compositions of many more solvents together. Okay, if it's possible, mobile phase contain more than 3 or 4 components should be avoided. It should limit up to the 3 only. Okay, because many times these mobile phase compositions, if you are using volatile solvents, may changes after a certain period of time if the solvent get evaporated from your mobile phase, and you will not get the proper separations, or you will not get the similarities, precision in your determinations. Okay, previous contamination of solvent, prevent contamination of solvents. Multiple components of mobile phase once used is not recommended for reuse. Okay, mobile phase साथ में फेंक देने का उसको. You have to use the fresh mobile phase when you are doing for the final separation process. Chemical reactions avoided between the stationary phase and mobile phases, that should be selected. Selection of mobile phase depends upon nature of the substance to be separated. Okay, so it has a role on the RF value. So what is the viscosity and polarity that will decide the sister phase RF value. The solvent used may be a single or a double phase system. So most of the time, what mobile phases which are using in TLC, same mobile phases we can go for the HPTLC with increasing polarities like N-hexane, cyclohexane, carbon tetrachloride, chloroform, benzene, toluene, trichloroethylene, chloroform, carbon tetrachloride is not chloroform. According to the increasing polarity, I have listed all these mobile phases here. You may note down this, or you will get these presentations if the organizers are giving it. Okay, chloroform, diethyl ether, ethyl acetate, N-butanol, acetone, ethanol, methanol, and water. Water is the most polar solvent which you are there in the mobile phase composition. Okay, which is avoided. Generally, we used to avoid the water. Again, a sample preparation step is there. Generally, we used to prepare the sample in methanol because it will get evaporated immediately, or chloroform mostly. So generally, methanol is used for HPTLC for sample preparation. For normal chromatography, solvent should be non-polar and volatile. For reverse-based chromatography, polar solvent is used for dissolving the sample. Sample and reference substances should be dissolved in the same solvent because we are spotting the sample and reference together to ensure compatible distribution at the starting zones. Okay, so sampling, how the sample is to be applied. Generally, if we are using the spots, sample are to be applied with small capillaries. Preparation of capillaries you may have discussed. Okay, we pull out the capillary for narrowing the end. नहीं तो उसमें से सबसे बारीक बारीक कैपिलरी यूज कर सकते थे नॉर्मल लेबोरेटरीज में, but in case of your HPTLC we are giving the bands, and bands normal के लिए हम लोग we can use the micro bubble pipettes, or we can have this capillary for the purpose. But in case of this HPTLC, micro syringes are used. ओके हमने जब इस्तेमाल किया था ये लिनोमेट 5 था, लिनोमेट 3। ओके लिनोमेट 3 के अंदर 3 4 5 तीनों भी मैंने आज तक इस्तेमाल किए हैं। तो लिनोमेट 3 के अंदर क्या था कि वो हम लोग पूरा उसको कहाँ सैंपल लगाने का है, कैसा सैंपल लगाने का है, तो वो मैनुअल करते थे। ओके इट इज नॉट कनेक्टेड टू द कंप्यूटर। लिनोमेट 3 पूरा सैंपल एप्लीकेशंस पूरा के पूरा मैनुअल होता था। जब हमने लास्ट टाइम यूज किया था वो लिनोमेट 5 यूज किया। तो लिनोमेट 5 में पूरा सैंपलिंग का प्रोसीजर एवरीथिंग इज देयर एज अ प्रोग्राम इन कंप्यूटर। ओके तो यहाँ पे क्या रहता है कि दिस इज द बेस वेयर द प्लेट इज टू बी फिक्स्ड। ओके सो ये प्लेटफॉर्म है, ये प्लेटफॉर्म मूव होता है। जनरली जब हम हाथ से स्पॉटिंग करते हैं तो हम हाथ मूव करते हैं और अलग अलग जगह पे उसको लेके जाते हैं। लेकिन अगर ये प्री कैलिब्रेटेड प्लेटफॉर्म है, इसका मूवमेंट पूरा प्री कैलिब्रेटेड है, तो जहां पे भी स्पॉट लगेगा, ये स्पॉट अभी ऐसे इधर लगने वाले हैं सामने। ओके स्पॉट्स आर इन दिस फैशन ओनली, ऐसे नहीं आड़े नहीं लगने वाले, ये मूव होता है और जैसे जैसे वो मूव होता जाएगा वैसे वैसे स्पॉट लगाते जाएंगे। Okay, so how it is moving, so it used to move in this horizontal way. Okay, so micro syringes are there, so we used to replace the syringes every time. After using, we have to wash the syringe and we have to keep it. Okay, so 1 microliter syringes are generally utilized. A very small spot is to be applied. Okay, this movement of the plunger is with the automated plunger piston. Okay, so simple syringe is there, इसमें कोई मैकेनिज्म नहीं है, सिर्फ वो प्रेस करने वाला मैकेनिज्म है जिसके अंदर वो प्रिसाइजली इट इज प्रेसिंग and the sample is getting spread as a spot, not as a spot, as a band of 6 mm or band of the 8 mm diameter, they are spread or spotted. Now same principle is there with the automated sampling devices. Okay, so this is the automatic sampler where we can have the variable volumes spotted. Okay, so this is automatic sample applicators. This is available with the Kamek laboratory. I've seen how it is working. Samples can be applied either as a spot or as band with this. Okay, preconditioning of the chamber saturation. Chamber saturation is very important part when you are doing. Okay, chamber हम लोग simple laboratory chambers use कर सकते हैं या उसका automated chambers भी available है जिसके अंदर plates रखा जाता है. Okay, sample risers बोलते हैं उसको. But उसके जगह पे हम लोग simple manual chambers we can use very easily. Okay, so time required for saturation depends upon the mobile phase. If you have saturated environment, then you will get the better separations. Okay, sufficient time should be allowed for saturation of your chambers. If unsaturated chambers used for development, the solvent evaporated from plate mainly by the solvent front, and it results in increased RF values. More time will be required, and RF value will be increased, and that is not supposed to do when you are doing the HPTLC. So twin troughs has been developed by CAMAG. Okay, so twin trough का मतलब क्या है कि simple हम लोग कोई भी chambers if we are using, so you require more quantity of the mobile phase to fill the bottom of the chamber. So here they have this twin troughs. Okay, जो center part है, so वो elevated center part है. Okay, so a trough of two troughs are prepared in the chamber. So one trough will hold at least 7 to 8 ml of the solvents. Very small quantity of the solvent is required. Okay. So what is the basic advantages of this twin trough? That at a time, two functions we can do with twin troughs. Okay. We can keep this mobile phase on one side of the trough and plate on the other side of the trough. Okay. So meanwhile, the chamber will get saturated with your mobile phase environment. The plate will also get saturated. Okay, and for development only you tilt this chamber so that the supernatant will move towards the other side and the development of plate will start. Okay, so this is a very easy way. Okay, so various sizes of plate holders or twin troughs are available. So gas phase conditioning with any conditions, liquids or volatile reagents can be done by using this twin troughs mechanisms. So again, various developing techniques can be used. So vertical developments generally we are using ascending chromatography that we are doing. Okay, vertical developments are there. Not to discuss too much because it is a routine practice of all our teachers, all your teachers working in the laboratory. We are well aware of these vertical developments. And what is a Varo methods of developments? That is a few new techniques you learn. Many people may know this Varo methods in which small grooves are there and which contain the different mobile phase at a time, and the plates is to be developed in this different mobile phase simultaneously, and it will optimize for optimization first step only. Okay, हरदम अपने को एक plate develop करना पड़ेगा. We have to develop individually the plates, then we have to optimize the solvent. Instead of that, we can have these Viro methods where all different solvent phases are kept into the different grooves, and at a time they develops and we can compare the spots. Okay, I will discuss this. And horizontal development method is there, and automated multiple development method is there. Okay, AMD methods. So these are the various HPTLC develop method. We can now see this Varo chamber development methods. In case of this Varo chamber development methods, so this is the developing chamber. In case of the developing chambers, we have these grooves which can hold the different mobile phases within them. Okay, so at a time 8 or 10 grooves can be prepared, and these grooves will develop. The plate is also grooved, or different strips are to be kept here. So we can have the comparison between the plates, between the spots developed by the different mobile phases simultaneously. Okay, so this is the very advanced technique of the developments, that is Viro chamber development techniques. Okay, development with 6 different solvents can be done instead of one time. Um, Sandwich as well as tank configurations can be stimulated side by side, making results directly comparable. Okay, different conditions of pre-equilibration indicating the relative humidities can be tested by using the simultaneous saturation methods. These variations of developing conditions can be freely combined in the single development steps. Okay, so again, the second rarely used is your horizontal development techniques, where the HPTLC plate is developed from both opposite sides towards the middle. Okay, so either we can use 10 by 10 plates or 10 by 20 centimeter plates. The plates are kept horizontally on the chamber in inverted position. जनरली हम लोग ऊपर की साइड पे रखते हैं प्लेट का सरफेस, यहाँ पे उल्टा रखना पड़ता है. The plate surface is below, okay, facing down. So HPTLC plates are kept into a chamber, developing chamber, which is closed with the lid, cover plates. Okay, then the mobile phase is moving from the side by using the certain wick type structure is there, wick like structure, which will take out the mobile phase on the surface. Okay, so and uh, glass plates, glass strips are there to hold these wicks in the center. Okay, and the development is conditioned, the trays are conditions. So last one is your automatic development techniques if you are using gradient illusion. But instead of using gradient illusions, that can be used for the isocratic illusions. Okay, I think most of you are knowing what is gradient and isocratic. Gradients, so mobile phase compositions remain same throughout the development technique, and in the gradient illusions, the mobile phase composition changes during the process of developments. It offers convenience, safety, and reproducibility for isocratic development and gradient development. Okay, the activity and pre-coating of the layer, chamber saturations, developing distance, and final drying can be presets and automatically monitors if you are having this AMD. Now, after the development, what are the pre-chromatographic steps? That is visualization, documentation, densitometric measurements. Okay, so these are the things which you can use. So visualization chambers, जैसे यूवी चेम्बर्स, यूवी कैबिनेट्स आपके रूम में रहते हैं, आपके कैबिनेट लैबोरेटरी में रहते हैं. सेम टाइप द यूवी कैबिनेट्स आर देयर. सो दिस इज मोर सोफिस्टिकेटेड, जिसके अंदर वो ऑब्जर्वेशन विंडोज है, जिसका अगर हम लोग यूज करते हैं तो इट विल prevent your eyes from getting exposed with the UV radiations. Okay, so such type of the UV absorption chambers are there. You can just locate your spots whether you have been doing it correctly or not, or it required the further developments. That all is decided by this UV cabinet. Then this is the scanner, densitometric scanner. Scanner 3 is there. We I use the scanner 3 only in the laboratory. So it can scan your sample in the UV range. It can scan your sample in the visible range. It can scan even the fluorescent range. Only the scanning lamp is to be changed. Either W lamp you can use, or you can use the tungsten filament lamp, or you can use the fluorescent lamp, or you can use the D2 lamps. Okay, so scanning simply done in this scanner. Different types of scanner is there. Okay, cost of the equipment is high, but recurring cost is low. Okay, one time cost is high, but if it get failed, then again the cost is high for replacement of the lamp. Then this is the photo documentation system as the digital camera of 2 Mbps, 2 Gbp, GB kya rehta hai wo. Okay, so there was available. Now our mobile photo cameras are too much powerful than this old camera techniques. अभी तो उन्होंने भी, they also has modified their cameras. Okay, but they are sensing macros. Okay, we have this macro setting in our mobile phones also. Okay, so they can if you are having this UV cabinet, we can place the UV camera or mobile on the surface, and directly we can take the photographs in the presence of UV without any flash systems. Okay, so same thing is there in the photo documentation department, in the photo documentation equipment. Here one more important things or difference which you can observe with HPTLC, that the simple plate after pre-washing are taken as a background image. Okay, so that is to be photo documented before we spot and before we developed. Okay, so that the background area can be substituted from your developed area. Okay, background substitution ke liye we had to take the photographs of pre-washed plate, and then it is to be placed into the instrument after development. Okay, so such type of the plates will get developed. This is the example of development. Adrenographs adrenographic paniculata plates, which can be seen by two wavelength, 366 nanometer and 254 nanometer. This plate is shown here. Many spots which are observed in the 266 nanometer cannot be observed in 254, but we can use both wavelengths for detections of various samples. Okay, each spot is responsible for the specific component of your extract. So you'll not get a single spot in case of the HPTLC. In TLC, we are thinking of the single spot. Here, we must get the multiple spots, because we are separating the components and we are identifying those components. But where to which part should be used for quantitative, that is your selection methods. Okay, if you are going for this spots for the quantitative estimations, the software, you have to add this area of your plates. First you have to observe And then you have to think of this. Okay, factor influencing separation of resolution of spot. I'm not going to discuss on these factors because one lecture only required for these factors. Okay, so type of stationary phases use, layer thickness, mobile phases, what you are using, polarity of this mobile phases, solvent use, light dissolves like solvent purities, what ionic composition of the solvent is there, that will decide the separation efficiency. Then size of developing chamber, saturations will required. Then sample volume to be spotted. Okay, size sample volumes. Too much concentrated spots will causes the tailing effect. Okay, here we can avoid the tailing effect because um, we know what concentration we can spot. Then size of initial spot, how much size should be there. Intermixing should be avoided. Keep the proper distance between the two spots. Then solvent level in the chambers, the baseline directly should not dip. Same principle might be there. The baseline should not directly dip into the mobile phase. Otherwise, you will not get any separations. Okay. Yes, friends, this was my first part, likely to be supposed to be finished within 1 hour. And the second hour, I was used to talk on this herbal drug standardization pro methods, but herbal drug standardizations and the separation or of the adulterants. Okay, so I was supposed to talk on both the topics, but I will summarize these parts because I can directly share you the documents, or you can share. I have shared directly the website. Okay, मेरे से ही लेने की जरूरत नहीं. You don't have to take from me. You can go by yourself on the website and you can download n number of literatures. But I can tell you, everything is available on the CAMAG website. Okay. Still, I will discuss this part. So when we are doing the herbal standardization, either we are doing the botanical parameters analysis or physical parameter analysis or certain biological parameter analysis or chemical parameter analysis, and we are using the different techniques to analyze all these parameters. If you are doing the botanical analysis, botanical, this is the common part which you are doing by your simple observations, that is either macroscopic or microscopic analysis of these parameters by using checking the color, order, taste, textures and fractures which are there. Okay, you use to fracture and you just find out the structure of the fractures to know the drugs. Okay, so up to that much skill you have to develop. Then what fracture and what is the surface of fractures will decides the identity of the drug components, drug herbal drugs. Okay, so microscopic is there, that is by your simple eyes or nose you can do auditory or textures of the surfaces we can analyze by your fingers. Okay, then botanical also includes the microscopic identifications that includes quantitative and qualitative analysis, powder analysis we are doing. Okay, so even how much powder many powder characteristics we are observing in the botanical analysis. SCM mostly used nowadays for this powder analysis, where we will get the more detailed analysis of your powder substances or the surfaces of your herbal drugs. Okay, for certain authentication purpose we are using SCMs. Then powder studies mostly we are doing by the biological analysis. So that part you are doing without use of any instruments. Then second, certain physical parameters, different instruments used for the physical parameters like your moisture content determinations. Okay, after harvesting, after cultivations, how much moisture is there. Okay, so that is also very important characteristic for your standardization purpose, because if more moisture is there, likely to get decomposed of the substance by the hydrolysis or for the microbial growth that causes the degradation of your sample. Okay, so moisture content analysis is the one most important part of your physical standardization method. Then extractive values you are doing. Okay, at what conditions you have harvested, because whenever we are getting the marketed product we don't know at what conditions they have harvested their plant. Okay, the harvested season, in which season they are harvested, because ready-made plants we are using from the market. So if you are doing your studies of your herbal drug, try to plant that herbal drug in your laboratory, in your environment. Okay, if the environment is suitable, you can plant a sample at your laboratory or at your premises. Then you can compare your sample product with that product. Okay, so that is again a double check for your sample. Okay, so there we are performing this extractive values, either water soluble extract, alcohol soluble extract, what are their solubilities in these solvents, that we are checking as per the monograph. Also monographs has been given for this herbal drugs also. Then ash values, either we are going for the insoluble ash or inorganic materials or water soluble ashes. Then we are going for the sulfated ash. Okay, so that is the physical parameters which you used to look out. Then fluorescence analysis we are doing for the fluorescent substances, secondary metabolite substances presence. Okay, so terpenes, terpenoids, phenols, which this type of this secondary metabolites has the ability to fluoresce in the fluorescent light. Okay, so they can you be studied by using these physical parameters. Then the biological evaluation, that is again important. What I am saying, that microbial contaminations, microbial load detections. Okay, so stability of your drugs or quality of your drugs is mostly studied by this microbial contaminations. Okay, then pharmacological evaluation for its toxicological studies. Many time the toxicological studies needs to be LD50. Okay, when we are doing this research, we have to need to find out the LD50 of your drug substances, what you have extracted. Then it is to be injected in animal also. Even if you are using the animal studies, you should have this LD50 data. Then and then only you are allowed to enter the substance into the animals. Okay, CPC committees are there, and they are asking you this LD50 data before you finalize your regimen for analysis of your any herbal drug. Okay, and the most important what we are looking is your chemical characterization in the herbal standardization by using chromatographic studies. Okay, TLC we are mostly utilizing. Then we can go for this HPTLC also for this chromatographic studies. The other chemical standardization may include your heavy metal detections, various techniques we can use for this heavy metal detections. That is, we have to detect this lead, cadmium, arsenic, mercury. Okay, so these are the agents which are reducing the markets of your foreign countries. Okay, many time even your drugs are effective, the contaminations of this will rejected getting them rejected from the foreign markets. Arsenic, lead, cadmium. Okay, um, we are having sufficient resistance for these substances, but their regulatory agencies does not allow such substances to be there in your plant material, and there we are lacking. So we should have sufficient research work. Okay, so one research work I have carried out, that was very much referred by the people. Shodhan Prakriya. In Ayurveda, there is a Shodhan Prakriya. Shodhan. So, Shuddhi. Shodhan, searching is, it is not searching. It is Shuddhi. Okay, so some chemical treatments leads to the Shodhan Prakriya, shodhaning of your sample, purification of the sample. And there we have to focus, and we have tried to focus of such things. One paper you may refer that paper, and later on I stopped working there because suddenly I lose this pharmacognosy touch. But even in the futures I may try for this shodhan prakriya. Okay, so there heavy metal detection is important. Then pesticide residues. The farmers are not knowing the use of pesticides in their area. Just wait. Okay, so this single slide will help you people for any standardization steps which you are taking in your laboratory. So now we will have some examples of this standardization and detection. Factor influencing separation we have completed, herbal standardization we have completed, applications related to the herbal drug analysis, that is simultaneous estimation of photo constituents in your sample, then quantitation of photo constituent in different varieties. That is also the area of your research, maybe that you are comparing the product which is there um, within the various physical areas. Okay, sources, different sources, you can collect the same material and you can analyze them for their constituent content. Simple. It does not need that you should go for the new survey or research. You can just compare the product which is available in the different areas. HPTLC can be used for determining morphological and geographical variations in herbal raw materials. Then evaluation and validation of poly herbal formulations. That is the major job HPTLC we are using for. That is poly herbal formulations. That is making its standardization very difficult. Okay, so documentary evidence for their content is very important. Used for determination of chemical constituents and its derivatives in bulk pharmaceutical dosage forms. Determination of adulteration, there we are using HPTLC, that our most important part of discussion. Important techniques for quality control of the herbal drugs standardization process. Bioassay guided isolation of plant antibiotics. Okay, certain plant antibiotics we can which are now developed can be studied. Bioassays of their can be done by using this HPTLC technique. Development of chromatographic fingerprinting for herbal drug analysis, drugs. Okay, fingerprinting means unique identifying characteristic for the drugs, that can be done by using this herbal drug analysis by HPTLC. Rapid structural confirmations and quantitation by, now this is the hypoenated techniques. I've seen this HPTLC mass equipments. It can also be coupled with HPTLC NMR, HPTLC EC. Okay, many hyphenated techniques are there. That is for the structural composition. Okay, not only that can be isolated from your extract, but that can also give you the information for their structural compositions or molecular structures. Okay, so in herbal analysis, this is a milestone. Okay, so this is HPTLC NMR. Hyphenated techniques, already we are very less know about HPTLC. Then we have to explore these areas for their um, advanced uses. Okay, so this validation of HPTLC for the regulatory methods, identifications. Okay, so one example is Garcinia indica fruits. The what we are using Garcinia. Anyone know? ऐसा मेरे को पूछना था. But as we, this is the one way communications. Garcinia indica, it is commonly utilizing as a kokum. Okay, so many time kokum, we know that which kokum is having the coca butters there. Which form of coca butter is very much useful, alpha beta. Okay, so characterization of this kokum, this HPTLC has given a great help in characterization of this kokum. So Garcinia indica consisting of dried pericarps of fruits of the Garcinia indica. Hmm? The fruits are well known in Asian countries and eaten for their health benefits. Okay, coca, kokum sarbat. So we are using this coca very well in every day. Okay, if what I can think in the laboratory, even if you do not have too many things to do. Okay, why I am shown this example to you, that with the TLC also you can show the extraction of the uh, this Garcinia fruits or pericarps, and you can prepare a colorful TLC plates and you can show to your students. Okay, even if you're not using the glass plates, you can call them to bring one HPTLC plates. and you can show their separations on this HPTLC plates. It's very easy to do this experiment. Even you can isolate this garcicinol, and it is not too much costly. Okay, you can have this garcicinol available in the market. You can use and compare your product. Okay, so you can do this HPTLC. Therefore, the first example was kept as a garcinol. You can see the separated components of garcicinol as a referrer or as your marker compound was spotted here. And this is the components: garcinia indica powder fruits, 100 milligram per ml, generally utilized. Garcinia gumin, the different varieties has been compared here. Gumin powder fruits and Garcinia Mosenta powder fruits. Okay, so you can compare the different varieties of Garcinia simply by using this HPTLC techniques that helps in the characterizations, as well as for the impurity profiling or the adulterant analysis. The second example for adulteration analysis is identification of herbal slimming drugs and screening for the adulterants by the HPTLC. Okay, if you see the market, health product are available. I will not give the names of any health product here. I'm having the restrictions. Okay, what cost they are taking from you, and whether they are giving, uh, you just compare all the health product which is available. Okay, so many Garcinia and some health products you are using for finding their contents. Okay, Garcinia, for obesity is the most important things. Today we are all worried about this obesity part, and we are purchasing tablets, 5 tablets, 10 tablets with a huge cost, capsules with a huge cost. Whether what type of adulterants are there, you can have your research product on this. Okay, many time when we are purchasing this, just I will show you one product I have purchased. ते बघ बरं product, ते वजन कमी करायचं प्रोडक्ट होतं ना तुझ्या ह्याच्यात, तुझ्या बॉक्समध्ये. Okay, even I am also obese and try to purchase this. Okay, Garcinia indica, Garcinia combegia. Okay, Neutralite. Oh, I don't have to give this name. Okay, so these products are available, and I found that it contain only the calcium carbonate with very small quantity of the drug inside, and they are giving 5,000 rupees for the 3 bottles. Okay, and that was also with the 50% 50% discount. And when we had done the analysis, we found that it contained only the calcium carbonate. Okay, so you can change your research topic towards this also. So Orlistant is a pharmaceutical drug designed to treat obesity. Okay, so with this herbal drugs, such type of the components are incorporated. Okay, which will reduce the intestinal fat absorption by inhibiting pancreatic lipase. Recently, some herbal stimulating drugs for weight reductions were commercially promoted. Okay, creating concerns in medical and scientific circles that there was a need to analyze samples of this drug for synthetic drug adulterants. They contain such synthetic drug adulterants that can be analyzed by HPTLC. That is a major research area in herbal. Okay, another such examples of the slimming supplements. Okay, see here this slimming supplement, uh, sabutamine, which was adulterated with the herbal drugs. 100% herbal, they are saying, and they are adulterating this drug with the these chemical substances. Okay, the market for herbal diets, slimming food products is vast and accessible for everyone, but it faces quality and safety issue due to the possible product adulterations and admixtures of illegal synthetic substances. Okay, whatever marketed products available, they get having these substances in it. Herbal slimming food supplements are not regulated by the regulatory authorities like drug safety rules. Therefore, illegal admixtures can be easily remain undetected and are a potential risk to the consumers. Okay, in that respect, you can think of your HPTLC research project. You can give this research project to your student, analyze these herbal products weight, because obesity is day by day increasing in people, and people are getting attracted towards such products. Herbal, they are safer. Even not in this, I have seen some examples of your homeopathic medicines, and these homeopathic medicines are also contaminated with the, or they are deliberately mixed with the steroids. Rheumatism, many people says that homeopathic medicines are more likely to use for rheumatisms, whether they contain any steroids. If they are analyzed, you will find that they contain steroids. Illegal process. Okay, so we botanists or we cognosy pharmacognosy people should start checking these drugs. uh, what will require, we require simply the marketed products and some standards. We don't require to go for the only separation of the drug substances from um, plant extract. This is also the most important literature or the research work we can do. Okay, so we can find out the potential risk to the consumer when they are consuming such type of the products. This is the another one. Okay, Vidania somnifera. So this is the example of Vidania somnifera by the roots and leaf with the HPTLC. Okay, so where USP methods are there. USP has given their important characteristics within a light. Okay, USP within a light. Um, so they are taken. Unknown origins they are found in the roots, powder roots of Vidania somnifera. These other constituents as their adulterants. Cannabis sativa. Okay, so again, this is the another drugs which gets uh, substituents of these drugs can be identified. Okay, the market, we can go for the exact species by just comparing the species which is available in the market. Cannabis sativa, the hemp plant, is one of the oldest crop of mankind, an important resource for the production of the textile fiber, food products, and medicinal drugs. And that is also getting adulterated with the various species. Even cafe, we can think cafe. Okay. Then separation of common plant triterpenoids by HPTLC. So we can separate out these uracolic acids, olenic acids, lipefin, lipefel acetates, lupol, by using this HPTLC tracking systems. Okay, you can see whatever various solvent systems which are used, and they are to be analyzed at the different wavelengths. Okay, so that is also means food. Those who are concerned with the food and they are doing food analysis, they can do this. Then identification of curcumoside extract according to the USP. USP has given this identification technique, curcumin. Okay, so curcuminoid is a practically purified natural complex of uh, dairyl lipinoid derivatives isolated from turmeric, curcumin longa L. Okay, curcuminide is a sum of curcumin, desomethoxycurcumin, and beside methoxycurcumins. The proposed method has been submitted for consideration in USP. This method is identifying curcuminide from curcumin longa by HPTLC fingerprintings. Okay, so all references are from Cramag. All for these slides have been well referred for their contents. Okay, so I got this all from Camac. Okay, another one, artemisinin in Artemisia annula leaves by HPTLC. This is another example of adulteration determinations. Powder decaffeinated green leaves. अभी सबसे ज्यादा हम लोग क्या कर रहे हैं? We are just going for the herbal tea. Okay, herbal tea. We used to find out the herbal tea. We are not drinking simple tea. Okay, we want herbal tea, and that herbal tea even get is contaminated with purified caffeine. Okay, because simply herbal will give a little stroke if you are again not getting proper. Even I have seen some most brands which are there in the lab market. They shows the contaminant of caffeine. We have find out one survey was done where we are just looking for the caffeine content of various herbal teas. They are saying they are herbal teas and how much caffeine they content. We have compared that caffeine content of various available marketed products, and we found that they get contaminated. Deliberation contaminations of the caffeine is there. Okay, and that can be do by HPTLC. Powdered decaffeinated green tea extract is prepared from the young unfermented leaves and leaf buds of Camelina sinensis konzu, also known as T. sinensis leaf. The extract contain no more than 0.1% caffeine, but many of the marketed products shows this caffeine contents. Okay, so we can go for the limit test. See, these are the limit test of various products done. Artemisia in the anti-malarial substances obtained from dried leaves of Artemisia annua, lin, to monitor the plant during the growth. So this is another one thing. We can monitor if you are doing the plantation in your laboratory and you are monitoring its growth, then you can use the HPTLC every time. Okay, so when to harvest the plant, what it contained, that can be shown here. This is the example of standardization of Ayurvedic formulations. Okay, the quality assessment of the herbal formulation is most important in order to justify their acceptability in modern system of medicine. If you want to make them official in our pharmacopoeias, we should give the documented evidence for their contents. Piperine, ambifilin, gallic acids, active ingredients can be detected in Ayurvedic formulations. Okay, such type of the areas pics we are getting and what area covered within this pic is to be studied as a quantitative tool. Okay, so this is standardization of the Ayurvedic formulations. Now, if you see the ginger rhizomes, Zingiberia officinale, okay, so these are the 15 components of this Ginginia officinale, and we can give this project to our young farm student to compare the ginger which is available in the markets. Okay, how we can find out this? So 6 gingeronal, 8 gingeronal, 10 gingeronal, 6 segol. Okay, so these are the rhizome extracts. They were compared with this 4 active ingredients. Okay, you can see these 4 active ingredients on the first slide. Okay, on the 366 and 254 nanometer wavelength, and the marketed product has been compared. So which market product shows which component more? Okay, so that can be studied here. Similarly, cinnamon variety derivatives, Sumatra. Okay, which cinnamons we are using? Sumatra cinnamon. The spices Cannamonium are useful as a flavoring part. Cinnamonium cassica has been adulterated in cinnamon verum. Okay, for their adulteration also, we can use this HPTLC method. Oh, too much time I have taken from you people. Just I will show you the examples now. This is the terminal archibula. Okay, in Triphala Churna, we are having the Herada Beherada. Okay, so this is the Beherada which is there in the termina tubula. Then uh, HPTLC in drug discovery also we are using. Then isolation of pure component having desired activity in time consumed and expensive. Okay, so this is another some examples of these drugs. Um, Identification of yerba mate, alkaloids from Adhatoda vasaka leaves. Okay, corticosteroids in herbal formulations that can also be studied. So these are various examples. N number of examples are available. Okay, as a time consents, I cannot. There was external link for this. I kept. See here, this PDF is available with the N number of the herbal analysis. Okay, 1 hour. It is even not sufficient to talk on single drug. This is the pharmacokinetic studies. Okay, madam, should we stop here?
Yes, sir. Yes. Yes.
Because lot many things are there to discuss, and I think it will be again more time consuming job.
Yes. Yes.
So these are certain references I should. PDCT HPTLC is there. Then this Sharma J, Fred B, Handbook of Thin Layer Chromatography, Bucket and Stilek. Various books are there. We can just refer those books. Okay, so I think I have taken your too much time in discussing all this part. Thank you very much. And the I will hand over the next part to the organizers. Now they can proceed with their things.
Yes, sir. Yes. Yes.
Yes,
Thank
I
you
will
very
also
much, sir.
start my camera.
Yes, sir. Camera. Yes, sir.
Yes, we can
Yes.
discuss
Thank,
now.
thank you, sir. Thank you very much for giving your new and valuable perspective on role of HPTLC in identification and purity checking of herbal drugs. Your thought-provoking sessions set a perfect platform for our researchers to do their best in the areas of this very challenging field of analytical science. So, looking into chat box, our participants has also benefited by the knowledge they have got from you. While the session was going on, some participants were asking questions regarding today's topic. We will pick a few from them due to time constraint. Shall we start our question answer session, sir?
Yes, sir, we can.
Yes, sir. Sir, first question is, can we use HPTLC for quantification of biological products like insulin or hormones?
It has certain limitations because in some it is related to some volatile products or some solubility parameters are there. So insulin has quantifications is very difficult by using a TLC or HPTLC methods. Okay, so there we go for the HPLC only. So that is again the area of research we can have certain more look out towards these things also insulin.
Yes. So next question is, which parameter should be considered for validation of HPTLC?
Yeah, area of the peak. Okay, so area of the peak and height of the peak. If you have a good marker available with you, you can compare that area which is produced by your substances and the area which is produced by your sample, and you can find out the parameters. Okay, validation if it are, then we have to go for accuracy, precision, robustness, everything of your method what you have developed.
Yes.
One more applications I am started with this HPTLC in Aurangabad, that impurity profiling of the drug. Okay, so that is also a major area where we can work with HPTLC. Okay, hyphenated HPTLC we can use to find out the impurities of your drug product, bulk drugs.
Yes. 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 Next question is, what type of the solvent system can be used in HPTLC for flavonoids?
Flavonoid contains, according to your flavonoids.
Yes.
Actually, I cannot give directly the solvent system methyl acetate, and because what are there, so you can go and use this Egon Stahl book. Even it is available with the library, or you can go for this CAMAC bibliography, where you will get the flavonoid, any sort of flavonoid separation. If they are doing the same separations, we can in mobile phase we can use with the laboratory also.
Yes. So next question is, can we use VRIO development chambers in our laboratory?
Actually, they are very difficult to get because they are very much useful. Actually, as we are using this synthesizer with 8 to 9 stations, such type of the VO chambers are available for HPTLC also. So if you have this HPTLC setup, you can definitely ask CAMAC to deliver such VO systems. And again, that is somewhat costly, but we can use.
Yes.
And you
So
can
next
think
question.
in-house
Yes.
production of or duplications of this type of the separation device at our end.
So next question is, can you please tell us something about HPTLC fingerprinting profile and isolation of marker compound?
Yeah, isolation we are doing practically also. We have done it practically also, because when we go to the market and we for 1 gram they are asking from 25,000 to 80,000 rupees. It was not affordable to us. Then what we have tried, we have prepared a thin layer chromatographic separations. We have scratched out that part of the thin layer chromatography. We have extracted that part, and we have checked it for the spectroscopic analysis, and then that that was isolated component was used as a marker compound in our laboratory. Curcumin we have isolated. Actually, it was easily available, but still for the practice purpose we have worked out with the curcumin.
Sir, one more question is there. What is the criteria for a selection of plates in HPTLC?
Depend upon what type of material you are separating. If you are looking for the simple separation, you can go for the normal adsorbent. But if you are having the drug component or substances which are requiring the reverse phase separation technique, then you can go for C18 plate. Okay, C18 plate is also available with HPTLC. So you can go for the C18 plates for separation of such type of the materials.
Sir, last one question is there. Uh, as a perspective of these herbal drugs, which chromatography technique is better, whether HPLC or HPTLC?
HPTLC we cannot use for the herbal drugs actually, because contamination is the major effect of the HPLC. Okay, no one will allow us to inject the extract or the separated component directly into the bulk drugs. Okay, so better we can go for the HPTLC, because at a time we have to analyze too many components together. So HPTLC will help us more than your HPLC.
Thank you, sir, and thank you all the participants. There are n number of queries from participants, but due to time constraint, we will stop the question answer session here.
You
But I
can
hope
do
all
one
the
thing. You can paste all the yes, madam. You can
yes,
paste
sir.
all the chart into one notepad file, and you can send that chart to me, so that even
Okay,
if it is possible, I can answer them by mail, or I can give you the answers, and you can circulate to them.
definitely, sir. Definitely. Thank you, sir. Thank you very much. Now it's time to present vote
Thank
of
you.
thanks. First of all, I would like to give a huge thanks to Dr. Uday Devkoti sir. Despite their busy schedule, they spare their valuable time with us. But I would like to request our assistant professor, Mr. Ramraj Omre sir, and organizer of this E-International Faculty Development Program to present vote of thanks. I would like to request Umre sir to please join us.
Hello. Hello.
Yes, sir, you are audible, sir.
I am audible to all of you. Very good afternoon to all of you. I would like to take this opportunity to propose vote of special thanks to today's speaker, Dr. Devkate sir, for his steering speech. I am extremely grateful to you, sir, to touch on the subject entitled Role of HPTLC in Identification and Purity Checking of Herbal Drugs in your speech and create so much awareness regarding the same. It would be our immense pleasure to invite you one more to spread awareness around the subject in the near future, sir. I also thankful from bottom of my heart to all the participants for their overwhelming responses to 1 week EFDP organized by our institute. I also gratefully thankful to the honorable management, to the organizing committee, to the convener, and to the coordinator, who is my best friend, Mr. Alkunte sir, for giving me such chance to propose the vote of thanks. Thank you once again to all of you. Thank you, Devkote sir, for being here with us. Thank you, sir.
Thank you.
I will hand over the mic to Chitra, madam.
Thank you, sir. Thank you for presenting vote of thanks. Uh, and thank you once again, Devpati sir. You have spared your valuable time with us and you have accepted our invitation and given your valuable time with us. Thank you, sir. Thank you very much. This is very knowledge boosting session for all of us. Sir.
Thank you.
I am presenting this virtual certificate. Please
Thank
accept
you.
it, sir.
Thank you very much.
It's a small effort, sir. Please.
Thank you. Yes, yes.
Thank you, sir. Thank
Definitely.
you.
Thank you.
Dear participants, tomorrow we will have our last day session of our International Faculty Development Program, and also we will have our valedictory function. So tomorrow we will have Dr. Jayaprakash Sangshetty sir from YB Chavan College of Pharmacy, Aurangabad, and who will orient us about quality by design in pharmaceuticals. So we will meet tomorrow at sharp 11 at morning. So stay tuned and have a nice day. Thank you.